Okay, so welcome back. You're in Bio 212, Anatomy and Physiology 2, and we're in Chapter 22. And what I'm doing is I'm going to go backwards a little bit here and pick up with exogenous and endogenous antigen processing, whereby an antigen-presenting cell, macrophage, something like that, dendritic cell, will either absorb, phagocytose, bring into the cell through endocytosis, an exogenous antigen, break it up into its smaller pieces, and then present that at the cell surface using MHC2. Okay? Make sure you pay attention to that. Exogenous utilizes MHC2. Whereas endogenous presentation, the antigen is already inside of the cell. And remember in the previous sort of discussion I said, well, that means maybe a virally infected cell will have something inside of it that can be presented at the surface of the cell via MHC1, but in both cases, these populations of cells are now antigen-presenting cells, where this little circle here is the antigen that will be presented to a T cell, a B cell, some other molecule. But you remember when I finished, I said, well, how does all this happen? How do these cells know to actually talk with one another? And like every system we've covered so far, there's going to be some endocrine responsibility. And here's the important thing. When we're talking about signaling molecules in the lymphatic system, we're talking about cytokines. So what I mean by that is cytokines are the hormones, H-O-R-M-O-N-E-S, that signal within the body. I know that looks messy, but I don't really care. Okay, and I don't mean that I don't care how you're reading it, but I want to make sure you understand we're talking about cytokines. Essentially, all the same mechanisms we learned back in Chapter 18 apply now. A molecule binds a unique receptor for that unique molecule, and that's how the body knows what's going on. They have specific names. Notice they're called interleukins, or tumor necrosis factor, or interferons. Each of these have different responsibilities. You're going to see there are a couple that I pay particular attention to, namely, say, interferons. These are a population of molecules that are made by virally infected cells to inhibit viral replication in uninfected cells. It's not the only thing they do, though. They also activate cytotoxic T cells and natural killer cells. They will stop cell division, and they'll stop the formation of tumors. Once upon a time, the families of interferons were thought to be the silver bullet stopping all disease states in the human body. And then, of course, they found out it wasn't the case at all. Signaling between T cells and B cells are going to require these interleukins, right? And you're going to notice when you start paying attention to their origin and function, who made them, what are they targeting? You're going to see the vast majority of cells here are T cells and B cells. Okay? So let's get into what cell-mediated immunity is. What we're talking about here is, can we identify a population of cells, namely T cells, that are going to identify an antigen to become activated? And the tricky part here is your textbook says a small number of T cells proliferate. Well, in reality, what's actually going on here is you have a cell, right? Let's go here. Let's go here. You have a cell. And let's call it a T cell, which means essentially it's primarily all nucleus. And on its surface, it has a receptor. Whoops, here we go again. I'm having those difficulties I had last time. You have a receptor on the cell, and that is specific to one and only one antigen. And that means over here, this T cell over here means it has a receptor, and it's looking for its one and only one antigen. And your textbook doesn't really make this clear, so you actually have to work this through inside your own brain. That also means that there'll be a population of B cells. Remember how your textbook drew those for you, large here. Probably should change the color here, so let's erase that. Boink. Now that'll erase the whole thing. I'm going to draw the B cell receptor so it looks something like this, right? And that B cell receptor has its own antigen, okay? But a subset of B cells and T cells have to see the same antigen. These are going to make antibodies against that one antigen. These T cells are going to make the receptor to bind that one and only one antigen, and these cells become activated. Both. Activated B cells are called plasma cells, right? Plasma. Activated T cells are called activated T cells. Act T cell. Okay? So generally speaking, it's pretty straightforward. Okay? So let's get rid of that. Erase the whole thing. Let's see if I can actually get this to become smaller now. How do I do this? Oops, other direction. Zoom out. There we go. Okay? So an antigen is recognized and bound. A small number of T cells proliferate. Think of it as one cell proliferates. It becomes clonally affected, and the antigen is eliminated. We'll see how that happens in a second. The first thing we're going to do is we're going to activate those helper T cells. Let's get this thing over here. Let's get rid of that. Here's a clonal T cell where you have an antigen presenting cell. Take a look at that macrophage. It's presenting that antigen here. And what you need to pay attention to here is there's that T cell receptor on its cell surface. And because it's a helper T cell, let's bold that, underline it, something like that to make sure you understand its significance. That helper T cell has a CD4 protein on it. And because the requirements for activation have been met, MHC2 is carrying that antigen that's activated that T cell receptor on an inactive helper T cell with that CD4 protein. It is now activated. Okay, whoops, put you back where you belong. Okay, this is, I think, what your textbook's trying to tell you. If that one cell is activated by that one antigen, it undergoes clonal selection and proliferation, and these helper T cells here, okay, are going to do something, and you're going to see what they're going to do. I moved that up here so you can see that they secrete IL-2. So that means activated helper T cells make an interleukin, and that's how you know that they're activated. Over time, once they've done their job, a subset of them, let's see, there are 5 of them here. Your textbook says there are 3 that survive. These are long-lived memory helper cells, and they respond more rapidly to the next infection using this antigen. Okay? It's not the only thing that happens here. Well, no, not at all. You're going to see that, let's get rid of this piece here. Let's go here. For us to activate a cytotoxic T cell, you'll see here that you have an infected body cell. You have that helper T cell that I can activate, that inactive cytotoxic T cell. And look over here. Here are the details. The cytotoxic T cell is activated by MHC1 complex and its antigen to activate that unique T cell receptor. And notice here, the cytotoxic T cell has CD8 molecules expressed on the surface of its cells. And having said that, what I want to make sure you understand is now that we've actually activated this, notice what was required. Remember that helper T cell a moment ago? And it made that interleukin? Notice it requires that interleukin 2. And when I lift this figure up here, notice what it's doing is it's running around inside the body now, and it's attacking infected body cells. Again, here's these 5 cells that are cytotoxic T cells that have done all that killing. Okay, a subset of them will survive, so they actually require less time to become activated by the time it sees the same antigen again. Okay. So what's actually going on here? What are they doing when they're running around the body? They are attacking that infected cell. It releases granzymes or granulysins and perforins. And what's happening, of course, is they're destroying the cell. Apoptosis is occurring, and now phagocytes come along and they eat that microbe. And you start the whole process over again. Because remember, now, whatever was inside this cell that required MHC1 is now being exogenously taken up, and now it's using MHC2. So now you can go back into this being an antigen-presenting cell and start the whole process over again until all of this pathogen has been removed. When you're looking over here, you're seeing that these are the molecules that actually create channels that allow the cell to undergo cytolysis. And again, we're going to have phagocytes come over and destroy the entire process. So again, now you can use MHC2 molecules to present that. How do we then shift? How does the body know that you can use antibodies to identify pathogen? And what's the difference? Well, one is a cell. That's the T cell. The entire T cell itself is quite large. Cells are large compared to proteins that are in solution of the plasma of your blood, which is what we're talking about here. And the way to think about it is all B cells are going to make some subset of antibody chains that are actually made up of 2 heavy chains and 2 light chains, and the antigen binding portion we'll get into in a couple minutes, but the antigen binding portion is what's binding the unique epitope by affinity, right? So all of these are unique to the exact thing it's supposed to be eliminating. Once you understand that, what I can do is say, well, if this is the case, what Mother Nature does is she has inactive B cells that have receptors on the surface of their cells. They're looking for unique epitopes, any which of could be activated by either a helper T cell for co-stimulation, okay, or direct binding to an antigen. Here, take a look at that microbe. They undergo proliferation and differentiation. And look at the difference here. What you're seeing now is, this is why we call them plasma cells. They secrete what was once purple up here as a soluble protein that's now traveling in the plasma of your body. And that's why it's called a plasma cell. Okay? A subset of these are going to die, a subset remain behind, and this is why you can make antibodies rapidly if you see the same antigen again. Okay? So the mechanism's similar, but notice the major difference is this. B cells make a soluble protein that will bind to antigen once released into the plasma of the cell, whereas T cells will attack or co-stimulate cells to ensure that they actually act properly, meaning remove antigen. So what is that antibody? This is an IgG molecule. This is a three-dimensional, space-filling model. And you can see the antigen binding sites are over here. Notice this cartoon is much prettier than the one that I drew you. But this is what I want you to see. You have 2 heavy chains. You have 2 light chains. You have variable regions, which will change what antigen will be bound to. You have constant regions that are actually used down here. The stem region down here, this actually draws macrophages to the site that the antibody binds to. So literally this acts as a homing beacon. Okay? So you have different populations of antibodies. You'll go over these on your own, but you can see there's G, A, M, D, and E. And even on your final exam review sheet, it says, look, you gotta know these. And notice there's a concentration difference. The most abundant is IgG, then IgA, then IgM, then IgD, and IgE. What happens, of course, is your body has this immunologic memory where once you actually see, let me reframe this here so it makes more sense. Once you see whatever that antigen is, okay, you can see that Mother Nature in the course of 14 days will create an increasing titer, and then after a second exposure, you can see IgM concentrations don't go up nearly as much, but you have this sort of long phase of production. But take a look at what happens when you see the second exposure for IgG. Take a look at the concentration. This is why you get your vaccinations, whatever they are. So the next time you see them, you actually have this really rapid response so your body can clear out whatever the pathogen is. Speaking of which, how do you actually get acquired adaptive immunity? There's naturally acquired active immunity, naturally acquired passive immunity. There's artificially acquired active immunity and artificially acquired passive immunity. When you're reading this, what I want you to do is realize that many of the autoimmune disorder states now, eczemas, sclerodermas, things like that, are actually treated by, take a look at the bottom of this figure here, intravenous injection of immunoglobulin into bodies. These are all pretty straightforward. This is the one that doesn't make any sense to the average human being. Most of your drug design, modern day drug design now, is what antibodies can we make that we can infuse into the body so it can remove whatever the pathogen is already present. Now these, both active and inactive or innate responses inside your body can be complemented. Did you pay attention to the phraseology? It can be complemented with another system inside your body. And I want to introduce this quickly. Okay, and that's the complement activation system that requires a series of soluble proteins that are cleaved and have different responsibilities. Notice C3 here is cleaved into C3b and C3a. B marks microbes. This is how macrophages know when a microbe is inside your body. But also C3a activates mast cells, which increase histamine, which drive perfusion of tissues to allow inflammation to occur. C5 is activated when C3 has been cleaved, and that C5 and C3 further enforce the mast cell activation, but also cause our C6, C7, C8, and C9 to create a channel inside bacteria. And that bacteria causes the bacteria to literally burst. This is called a membrane attack complex, and I'll get into that in class. Okay, what does it actually mean when you make millions of these on the outside portion of a cell? The last topic we need to talk about probably should have been at the beginning of the text, and I don't know why they don't put it at the beginning of the text, but how does all of this work? T cells have to undergo the ability to have a positive and a negative selection to make sure that self-MHC antigens can be seen. But take a look at what's different here. Do immature T cells recognize self-MHC? And if the answer is no, it means you have to destroy them. If the answer is yes, it means they should not be able to see self, because if the answer is yes, they have to be killed. Okay? They should not be able to see self, and this is actually how you make T cells that can recognize MHC molecules. They then move to the lymphatic tissue inside your body, and they become activated by antigen and co-stimulation. If these others happen, bad things tend to happen inside your body. Most of the autoimmune disorders I mentioned just a moment ago seem to occur probably as a result of anergy, of the improper activation of T cells. I can't speak specifically to what the mechanisms are for all of them. If you have specific questions, I can definitely do some digging around for you to explain them better. But, these cells should be killed, but not all of them are. How's it different inside B cells? B cells development, same thing. They are immature inside the bone marrow. They have to be able to see, mature B cells should be able to see self-MHC molecules. Okay? And then they should be able to get a co-stimulation, which is then going to differentiate into plasma cells, and they're going to release antibody. They should not be able to inactivate B cells as a result of inappropriate signaling. You don't want to have excess antibody inside the circulatory system if you can help it. So these 3 videos pretty much were an introduction and description of a little bit more detail to help you understand why your textbook put things together the way they put them together. The next set of supporting videos, probably 2 of them, will have review questions for the 3rd exam. Okay? All of chapter 22 will be found inside of those. Okay? Have a good night. Be safe. I'll see you guys in class.